students, welcome back. In mental study, we gain knowledge about the characteristics that are controlled by a single gene that has multiple alleles. For example, the height of a plant is determined by either capital T or small t. If the plant inherits the dominant capital T, it will be tall, and if the plant is dwarf, that means it inherited small t. Is it possible for a single phenotypic trait to be determined by multiple genes? Yes, if you take a moment to observe your surroundings, you will discover characters that have wide range. An example is the height of human beings. Humans come in various heights, not just tall or short. There is a broad spectrum of potential heights. That means the trait human height is controlled by more than one gene. Let's illustrate this through another example. The human skin exhibits a diverse array of colors. There are several shades, some being darker, others lighter, and some falling in between these two colors. That means the trait human skin color is a polygenic inheritance that are controlled by more than one gene. Each allele in each gene contribute towards the human skin color. Let's assume three genes control the skin color, namely capital A, B and C. Gene A has two alleles, capital A small a. Gene B has capital B small b, and gene C has capital C small c. These capital ABC add the melanin factor which gives color to the skin. Small abc have no melanin. If a person's genotype is capital AABBCC, the skin color is very dark as all the alleles are dominant and they add the melanin. The person with genotype small AABBCC has very less melanin and his skin will be very light. Other combinations will be in between these two colors. We know that a lot of combinations are possible like capital A small a, capital B small b, capital C small c, and capital A, capital B small b, capital C small c, etc. The people with more dominant alleles will be dark and the people with more recessive alleles will have lighter skin color. Now let's learn about a new term, pleiotropy. In Mendel's experiment, we observe a single phenotypic character is controlled by a single gene. Polygenic inheritance demonstrates that multiple genes are responsible for a single phenotypic character. Pleiotropy is the exact opposite of this. Sometimes one gene is responsible for more than one phenotypic character. This gene is called pleiotropic gene and the mechanism is called pleiotropy. Let's grasp this concept using an example. Phenylketonuria is a disease in humans caused by the mutation of one gene. Gene mutation means a change in the base sequence of DNA in a gene. We will cover the topic of mutation later in this video. Phenylketonuria is caused due to mutations in both the alleles of a gene that codes for an enzyme named PAH, phenylalanine hydroxylase. This PAH converts an amino acid named phenylalanine to another amino acid, tyrosine. Tyrosine helps to produce melanin, the pigment responsible for hair and skin color. It helps in the function of organs responsible for making and regulating hormones, including the adrenal, thyroid, and pituitary glands. It is involved in the structure of almost every protein in the body. If mutation happens in both the alleles of the gene that codes for PAH, the enzyme becomes inactive, so phenylalanine cannot be converted into tyrosine. Two problems occur here. The concentration of phenylalanine increases in the blood, that leads to toxicity, and the second one, tyrosine is not produced. The absence of tyrosine leads to the occurrence of numerous phenotypic changes like mental retardation, hair loss, and skin pigmentation. Tyrosine is essential for melanin production. Without the presence of tyrosine, the production of melanin will cease and it results in skin whitening and early white hair. Here we saw that one gene is responsible for multiple phenotypes. That is called pleiotropy. In ancient times, scientists were puzzled by the mechanism of sex determination. It was the German cytologist named Henkin was the one who shed some light on this puzzle. While doing some experiments on several insects, Henkin made an intruding observation. The presence of a particular nucleus structure throughout the process of spermatogenesis. Spermatogenesis is the process of developing sperms. Henkin also noticed that after spermatogenesis, only 50% of sperms received this nuclear structure and other 50% didn't. He named it X body, but he could not explain its significance. Further studies by other scientists revealed that this X body is actually a chromosome and they named it X chromosome. Now let's understand different types of sex determinations. Let's first discuss XO type of sex determination. This type of sex determination is found in grasshoppers. The male grasshopper produces sperms in which 50% have X chromosomes and other 50% of sperms do not have X chromosomes. That means the male is XO type. Some sperms have X chromosomes and others don't. O represents the sperm that lacks an X chromosome. However, in female, the eggs have two X chromosomes. Let's draw a Punnett square for sex determination. When XX and XO combine, there is a 50% probability that the resulting offspring will be either male or female. When the sperm carrying an X chromosome combines with the egg, it will result in the birth of a female offspring. If the sperm that lacks an X chromosome merges with the egg, the result will be a male offspring. The male grasshopper has one fewer chromosome than the female. Now let's understand the XY type of sex determination. This is found in a number of other insects like Drosophila melanogaster and mammals including humans. We know that there are 23 pairs of chromosomes in humans, 22 pairs of autosomes and 1 pair of sex chromosome. In males, there is 1 X chromosome and 1 Y chromosome. This Y chromosome is distinctively smaller when compared to X chromosome. In females, there are 2 X chromosomes. Let's observe the Punnett square. When an egg and sperm unite in fertilization, there is an equal 50% probability of the resulting offspring being either male or female. In each pregnancy, there is 50% chance that the child becomes a male or a female. If the father's sperm fertilizes the egg carries an X chromosome, the child will be female, whereas if the sperm carries a Y chromosome, the child will be male. As male produces 2 different types of sex gametes, X and Y, this phenomenon is known as male heterogamety. Exotype can also be called as male heterogamety as males produce 2 types of sperms, some with X chromosome and some without X chromosomes. In birds, we can see the reverse of what happens in humans. Here, males have a pair of Z chromosomes and females have 2 different sex chromosomes called Z and W, ZW type. There is another type of sex determination mechanism called Haplodiploid sex determination. This is found in honeybees. There are mainly 3 types of honeybees: queen, worker and drones. Here, the queen and workers are females and drones are males. Let's see how we can determine the sex in them. In honeybees, the females are diploid with 32 chromosomes and males are haploid with 16 chromosomes. In other words, females are formed by the fertilization of eggs and sperms. Haploid egg and haploid sperm unite to form diploid zygote. These are the females. Males are formed from females without the fertilization of eggs through a process called parthenogenesis. The egg itself developed to form an individual without fertilization. The egg is haploid, so the individual that formed from egg without fertilization will also be haploid. Hence, the male honeybees don't have a father as they are formed without the contribution from sperm, but they have a mother as they are formed from the queen. However, the queen and the workers have fathers as they are formed from fertilized eggs. Hence, we can say that male drones have a grandfather, the father of queen, but have no father. We have just discussed that queens and workers are both female, but what sets them apart? There will be only one queen in the hive. When the existing queen dies, one of the female larvae is fed with special royal food and a queen develops from it. As we discussed earlier in this video, mutation is a structural change of the DNA sequence of a gene. Mutation is an extensive topic, but let's try to grasp the concept of mutation in a simplified manner. We know that DNA is in a highly coiled form. If any part of the DNA gets deleted or inserted to the original sequence, there will be a change in the chromosome. This is called mutation. Here you can see a portion of the DNA is deleted from the sequence, which is a deletion mutation. Another example of mutation is inversion, where the portion of DNA becomes inverted within the original sequence. Here you can see DEF is inverted here as FED. And this is the sequence of DNA where the nucleotides are inverted. There is another mutation called point mutation. A point mutation occurs in a genome when a single base pair is added, deleted or changed. We know that DNA is made up of base pairs like adenine, thymine, cytosine and guanine. Here, adenine can be paired with only thymine and cytosine can be paired with only guanine. For the sake of comprehension, let's construct a DNA sequence CCT-GAG. They will be paired with GGA-CTC. Here, the RNA formed will be CCU-GAG. You might be aware that the 3 nucleotide code for one amino acid. GAG code for an amino acid, glutamic acid. But due to mutation, this T can be replaced by A. Hence, the sequence will be GGA-CAC. If thymine is replaced with adenine, then the other strand will also change accordingly. Here, instead of adenine, thymine will come. Then the RNA sequence will also change accordingly. For example, GUG instead of GAG, because uracil in RNA pairs with adenine. Hence, the triplet code will change to GUG from GAG. GUG codes for another amino acid, valine, instead of glutamic acid. Valine causes the hemoglobin molecules to adhere and create elongated fibers within the bloodstream. This condition causes a distortion in the shape of red blood cells, turning them into sickle-shaped. Sickle-shaped red blood cells get stuck in the walls of small blood vessels. We know that hemoglobin carries oxygen molecules. Sickle cell anemia is caused when certain body parts don't get enough oxygen because they are trapped in the blood vessels. Mutations can change the structure of DNA sequence, which is a genotypic change, and the disease that happens is a phenotypic outcome. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.